So let me show you an example now of an existing model of tumor invasion, which was developed by Cohen et al. So here is the Boolean model that we are going to that I'm going to show next. So as you can see, you have a lot of nodes, around 20 or something. I forgot the exact number. And we have two important types of nodes. The first one will be the input nodes. They are, like the node A in my simulation, nodes that will stay constant during the simulation and that you set at initial time. And so here we'll have two input nodes, one called EC microenvironment, which will basically describe the influence of a microenvironment on this system of tumor invasion. And the other will be the presence or absence of DNA damage. This can be this DNA damage can be caused by some drugs, for example. And we have another type of nodes, which are the output nodes. These are the ones which will be like the readouts of our system to be able to see what really happened at the end of a simulation. So here we have different output nodes, EMT for epithelial mesenchymal transition. We have cell cycle arrest, which will be true if the cell cycle is arrested. We have also a node for apoptosis, which will say that the cell will be activating apoptosis. We have a node for invasion, which will be that the cell starting the invasion process. Migration, which means that the cell will be migrating, and metastasis, which is basically similar to migration. So if we simulate this model, we obtain, with the default simulation, we obtain this final state distribution. So we have mainly apoptotic cells which have stopped their cell cycle, around 43%. We also have around 30% of cells which are invading. We are 20% of cells which are at homeostasis and will keep proliferating, basically, because they are not, their cell cycle is not arrested. And 6% of cells where their cell cycle arrested. So one important aspect of this, what I call the default simulation of this model, is what are its input nodes. So I presented you here the two input nodes, EC microenvironment and DNA damage. And in this case, what we do with WebMaBoSS is that we want to be able to see all the possibilities of the model, whatever their initial values. So what we do actually is that we set the initial values like I explained to you quickly before. We set it to random, which means that 50% of the time this will be false and the other 50% will be true. And same thing for DNA damage. And so actually what we get is that 25% of the time both will be off, 25% of the time one of them will be off, and the last 25% then both of them will be on. So doing this, we're able to really sample all the possible initial values and look at some kind of global behavior of a model. But we can also just choose some specific initial values to be able to simulate the model and see better like what will happen in certain conditions. So here, for example, we want to simulate first with no microenvironment and no DNA damage. So both input nodes will be set to 0. And then we see that most of the cells will keep proliferating and that 18% of the cell will block their cell cycle. But then if we do another simulation, this time activating the microenvironment, the influence of the microenvironment, but still not with DNA damage, we can see that we get a completely different phenotype. We get only invasion and cell cycle arrest, meaning that all the cells will be invading and turning into metastasis. What we show here is that when we activate the microenvironment, when we set this input to 1, we obtain an invasive phenotype 100% of the time. So now what we can do is modify again the initial values. So we keep the microenvironment on, and this time we will add DNA damage. And so again, DNA damage is something which can be triggered by some drugs such as checkpoint inhibitors. So this is something which is used in clinics. And so here, when we add this input to 1, what we can see is that 80% of the cells now are turning apoptotic, cells are dying. And only 20% of the cells will keep being invasive. So we are able, for example, with this treatment, which activates DNA damage, to kill 80% of the cell in the tumor. And so, just also to explain to you again, so when we see here the wild type, which is the first simulation I showed you, the default simulation. So in this case, if we simulate all the combination of the 4 different values for input, and we basically sum and look at the probabilities of with these 4 inputs, we will get exactly this picture. And so here, what we want to do then is to simulate a mutant. So forcing the node to be active or inactive. So in this case, the mutant we want to simulate is we want to force the activation of Notch inside our pathway and force the inhibition of p53. So we start again this time from this wild-type simulation, this default simulation, where we have all the possible inputs. And what we see is that when we simulate this specific mutant, now again we get a completely invasive phenotype. So this is one of the known tumor, which this has been shown experimentally. Then when we have these this double mutant, then you get a lot more invasive phenotypes. And so that's something that the model is able to reproduce. And so now let me show you briefly how to be able to reproduce this, this example with WebMaBoSS, and then we'll take a break and actually do it ourselves. So first, what is WebMaBoSS? WebMaBoSS is a tool we developed, which is a web tool, so that everybody is able, without installing anything, without too much knowledge of computer science and scripting and programming, people are already able to test MaBoSS model and simulate them and do some funny things with them. So here is the first page of when you arrive on the website. So we have some demo projects which are available, as well as some tutorials. But the best way to use WebMaBoSS is first to create an account. With this account, you will be able to have some kind of repository for your models. You will be able to download some models there and keep them in this repository and simulate them, and also save the simulation to be able to reaccess them later. So it creates a kind of personal space for WebMaBoSS. So to register, it's easy. You just have to click on register here on the top right, choose a username, email is optional, and choose a password. I think most people already did, but if you didn't, please do. And then you just have to sign in with your username and password. After creation, you don't have to activate your account or anything. It will immediately work. Once you are connected, you will have a list of projects. You have, I think, 1 default project when you create your account. And so here you have this repository, like I told you, which is divided into projects. So you can just create a project and put some models in it, but also create more projects so that everything is not in the same place. So to create a model, just click to create a project, just click new project and choose a name and a description. And then once your model is created, you just click on the project, and you will arrive at the models page of your project. So here you are able to load or import models into your project. So first loading, you just have to give it a name, select the type of model. So here we will have an example where we use MaBoSS type, so the BND and CFG files I showed you before. But this also works with GenSim, BNet, and SBML qual models. But you also have the options to import models from existing models from databases. There are 2 big databases for logical models. First one being biomodels, and the second one being cell collective. And so by clicking on the import model, this dialog will show up, and you can just select one of these models, click, and it will download it and inside your list of models. Then once you click on the models, we have some representation of a graph of interaction of the model. So this is not exactly pretty, but you can still move the nodes and be able to organize it by yourself, or choose automatic algorithm to organize them, to make the layout. But you can already use it to look at which influence has this node on the other and this kind of thing. Then you have a simulation part where you can launch simulation, and you just have to click the new simulation button, and it will open this dialog. So here you can put a name of your simulation. If you want to save it and reaccess it better later, it might be better to put an informative name, but it's not compulsory. Then you have the maximum time of the simulation, the sample count. You have some settings such as discrete time that I didn't told earlier, but if you want to stay closer to the Boolean formalism and not have continuous time, you can switch to discrete time. And other settings which are more technical. Then you can set the initial states of the system. So here we have some kind of sliders where for every node we can set the exact percentage of activation for this node. So what we want, for example, if we want to reproduce the default simulation that I showed you earlier, is to have these two input nodes, DNA damage and EC microenvironment, at 50%, so that they will be active and inactive half of the time. And so you will be able to sample the initial states. We also have, like I said, the notion of output nodes. Again, you don't want to have all your output nodes active, otherwise you will have just too much states because of combination. So you just keep some of them active to have results which are easy to read. And in this case, basically, we put the output nodes. We choose to have the output nodes of a model as output of a simulation. And finally, we have a tab about mutation, which allow you to perform mutation for your simulation. So here, for example, we can see that most of the nodes are not mutated, but AKT1 here will be forced to be active. And you can click on the left part of the slider, and AKT1 will be forced inactive. Once the simulation is done, you open this window where you can see the result. So first, you have the final state distribution, like the pie chart that I showed you already a lot of time. And so here we see basically the same result as I showed you earlier, where we have mostly apoptotic phenotypes, some invasion here, some cells which are keep proliferating, and a small percentage where the cell cycle is arrested. We can also look at the trajectories. So here will be the trajectory of nodes, where we can see the different nodes. We cannot see the bottom of the figure, sorry, but we will have a list of nodes at the bottom, and you are able to click on them to plot them or not. Same thing with states. And we also have the list of fixed points. So fixed point, I didn't introduce it properly, but it's states of the system where no evolution is possible anymore. So when WebMaBoSS is simulating, it's also tracking these states to be able to list all the possible states of the system, which are accessible from the initial state you're simulating, where the system doesn't evolve anymore. And so here we have a few, here 9 steady states, and you're able to click on this blue button to be able to see the interaction graph with the nodes green or red, depending if they are active or inactive in this fixed point. One important thing with WebMaBoSS is that you're also able to edit the model. So you can just download them and edit them. So here we have the parts to edit this activation and inactivation rates. So you have a list of nodes, and then you can open one node by clicking on the blue button here. this will show the logical formula which is used and the activation and inactivation rates in exactly the same way I presented you before. And so if you want to edit them, you're able to just click on this button here and modify, for example, here the logical formula of which is used then in the activation and inactivation. And when you type these formulas, you also have some error checking mechanism to show you if the formula is valid or not. You can also edit initial values for your model. So here is exactly the same screen as in the simulation. But this time, again, if you are in this editing part of WebMaBoSS, this result won't be only for one simulation. You will modify the model, and the model will stay that way. You also have, again, output value to be able to define which nodes you want to plot in your simulation. Parameters, so like it can be parameters which are used for the kinetic rates to activate or inactivate the nodes. And finally, settings. You have here all the possible settings of this MaBoSS simulation with the max time, the sample count, the time tick, so the interval between each time point of a simulation. And finally, one possibility of WebMaBoSS is the possibility to do sensitivity analysis. So sometimes what we want to do is we don't know exactly which mutant will be able to work with a specific phenotype. So we just want to try a lot of possible mutants and see what works. And so this is what this part of WebMaBoSS is for. So here again, you can select a name, and but it's not compulsory. And you can choose to do either you can choose to do single mutation or double mutation or both. So single mutation will be that for all the candidates that you selected, and this I will show you a little later, you will modify only one node at a time. And then if you click on it, you will see that if you activate this, you are able to choose if you want to have only inhibitions or only activations. And so you have the exact same thing for double mutation, but this time it will do a lot more simulations because it will try every combination of candidates. So then you have a tab to select all the candidates, so all the nodes that you want to mutate. So here we have to be reasonable because, again, like if we try all the double mutants, this will mean a lot of combination of candidates. So which will mean heavier computation and a lot of time to actually get the answer. So in the example we'll do today, we shouldn't tick too much, like 4 or 5 will be already enough to have a quick answer if we do double simulations. But no, I think the example will be single simulation, if I remember correctly. And then you can select a little more candidates. I think all of them should be enough if we stick to only inhibition. And in the example I'm asking you, it will be only inhibitions to simulate. And then we also can choose the output because, again, we have to select some nodes, not all of them, to be able to have results which are simpler to analyze. And so once you run this sensitivity analysis, this is the type of result you will have. So here I tried all the single mutants only inhibition. So you can see that I tried the inhibition of AKT1, AKT2, CDH1, CDH2, etc. And so you will have a big list of this individual simulation, basically, of this mutant simulation. And what you can do if you want to look for a specific phenotype is to click on this filter button here on the top right. And here you're able to filter the simulation results. So say, for example, I want to select all the simulations which have this invasion cell cycle arrest phenotype inferior to 20% of the time. So here we put 0.2 because it's its probability. And once we do this, we obtain only a few of these phenotypes.